Okay, now let's talk about new stuff. Lipid metabolism. So the first unit that we're going to cover the biosynthesis of fatty acids and triglyceride. So now major lipids in the body, I guess you guys all know, right? What kind of lipids that our body has. First, the lipids present in the whole body, in the plasma circulation, and also in the tissue cells, right? The plasma and tissue lipids includes the fatty acids, monoacylglycerol, and diacylglycerol and triglycerol, right? And also we have cholesterol esters, phospholipids, and sphingolipids, and other intermediates of lipid metabolism. And in terms of sources of plasma tissue lipids, where do these lipids come from, right, in our body? So actually, two main sources, similar like to the carbohydrates, right, or proteins. We have lipids that come from the diet, and our body can also synthesize lipids, right? So two sources: dietary lipids and synthesized lipids, right? And what is the role of the lipids? Why we need lipids? What does the lipids do? So first, the energy source. Right? We know the triglyceride, mainly that's stored in the fat tissue. It can be break down to fatty acids, and fatty acids we know is the energy fuel, right? The substrate, energy substrate. So fatty acids can be utilized, oxidized in the tissues, and for ATP production. So this is the important function of lipids. And lipids or fat oxidation actually is pretty efficient way, right, in terms of energy store. For example, if we burn 1 gram of a fat, we're able to generate 9 kcal of energy, right, compared to only 4 cal energy per gram of carbohydrate or protein. So fat is pretty efficient, right, energy. And also when we stored a 1 gram of fat, we only needed 1 gram of water compared to the 4 gram of water needed for store 1 gram of carbohydrate or protein, right? So it's very efficient way to store energy, and secondly, lipids is also very important for the structure component, right, of the membrane, the cell membrane, and also organelles, right, inside of cell. Lots of organelles like mitochondria or ER or other Golgi, other organelle membrane, or need lipids, right? The lipids form the bilayers of the cell membrane and also organelle membranes, right? So lipids, is very important. And those organelle or lipids, the membrane lipids, usually are the acylglycerol, phospholipids, sphingolipids, and cholesterol, right? So all these membrane, lipids are important. And thirdly, lipids also serve as so-called lubricants, right? We need a lubrication or conditioning of our body surface, right? So lipids also present, the sebaceous glands, right, on the skin surface. And lastly, now we know actually some of the lipids are very important, serve so-called signaling molecules. So that means some of the lipids or metabolites of lipids that can regulate metabolism, can regulate some enzyme activities or gene expression or stuff, can regulate the body or cell metabolism, right? So, and lipids can also be used for synthesis of sex hormones and steroids, right, or vitamin D, right? So these are the functions or roles of lipids. So we need lipids not only just for energy, but also for compose, the membranes and also signaling molecules, right? And lipid synthesis, I would like you to make sure that you understand actually the two different concepts, right, in terms of lipid synthesis. First, the de novo synthesis of fatty acids, we also call lipogenesis. So de novo synthesis of lipids or fatty acids or lipogenesis is different from the other pathways we call the triglyceride synthesis, right? So de novo synthesis of fatty acids or lipogenesis is basically it synthesizes fatty acids from the acetyl-CoA molecules, right? So again, the lipogenesis or de novo synthesis of fatty acids is a process that synthesize fatty acids from acetyl-CoA molecules, right? And triglyceride synthesis, can you guys describe the process? How, triglyceride synthesize? What are the substrate of triglyceride synthesis? Triglyceride, see here the structure of triglyceride, glycerol backbone, and fatty acids, right? So again, the substrate of triglyceride synthesis requires two substrates, right? Glycerol backbone and 3 fatty acids. So the process is a synthesis of triglyceride from glycerol backbone and 3 molecules of fatty acids, right? So it's actually a different process, right? Lipogenesis versus triglyceride synthesis, right? To make clear, these are the two different processes. Now let's talk about the lipogenesis, so de novo fatty acid synthesis first, right? This pathways or process. So what are the primary tissue for lipogenesis? What do you think? You can take a guess. Yes, great. Liver, and what else? What else? Can do, adipose tissue. And think a little bit more, we have a third tissue that also can do the lipogenesis. Muscle, right? So again, the liver is a primary tissue that does lipogenesis. That means lipogenic activity is very high in the liver. And the second tissue, the adipose tissue. But compared to liver, lipogenesis activity in adipose tissue is relatively small. Relatively low compared to liver. Again, liver is the primary tissue for the lipogenesis, and then the adipose tissue. And muscle can also do some fatty acid synthesis or lipogenesis, right? So here I just put the two main tissues, right? Liver and adipose tissue are the primary tissue for lipogenesis, but I also wanted to be aware that muscle can also do the fatty acid synthesis or lipogenesis. And how high-fat diet will affect or regulate lipogenesis? What do you expect if we eat high-fat, very high-fat diet? What do you think? Lipogenesis should be decreased. Why? How?
Because one of
Mm-hmm. Okay. Well, yeah, I think your first answer is right. Great. It's actually high-fat diet suppresses or decreases lipogenesis, right? And in terms of how, we're going to talk about later, right? Thank you. And then how about, okay, so high-fat diet basically inhibits or suppresses lipogenesis, right? And keep in mind the question why, how high-fat diet suppresses lipogenesis, right? We're going to talk about later the sort of mechanism, right? And next, how about a high carbohydrate diet? Increase, yes. Increase or stimulates lipogenesis. Yeah, I think you did mention that, carbohydrate and, right, acetyl-CoA then can stimulate the lipogenesis. When we have a higher carbohydrate diet, we, get very active glycolysis and produce a lot of acetyl-CoA, right? And this will serve as the substrate for fatty acid synthesis. If you still remember the TC cycles question?
Yeah.
Yeah.
How high fat, how high carbohydrate? Like, how are they defining high fat, high carbohydrate?
Oh, that's a great question. So that means when the carbohydrate, for example, the normal diet, right? We usually have a certain portion or percentage of carbohydrate in the diet. If the carbohydrate is higher than the normal, percentage in the diet, then we'll consider the high carbohydrate diet, right? And similar, for the high fat diet, right? For example, like when we doing kind of an animal experiment, right? We try to feed the animal or mice a high-fat diet. We usually, the high-fat diet, the fat contains 60% of the total energy. So, this kind of just relatively high. Okay, so the last point, what I'm trying to explain is that like Western diet, basically I think we'll consider the high-fat diet, right? So it's not a significant contribute to triglyceride synthesis. That means when you have a high-fat diet, then basically high-fat diet inhibits the lipogenesis, right? So lipogenesis will not be very active when you eat a very high fat diet, right? So in other words, that point is lipogenesis is not actually significant contribute to triglyceride synthesis when we eat a high-fat diet, right? Does that make sense? And basically just, simply just remember the high-fat diet will inhibit lipogenesis, right? So we're going to talk about more later. Okay, so now the concept. What is the purpose of lipogenesis? Why we need to do lipogenesis? First, lipogenesis we described, right? The concept. The definition is the process that converts surplus carbohydrate and protein to fat and stored as triglyceride in adipose tissue, right? So this is a process that is defined as the lipogenesis. So I guess you guys have a right answer when we did sort of background test, right? I think one of the questions asking whether carbohydrate or protein can be converted to fat, but your answer, most of them, you answer yes. So this is something, related to the lipogenesis, right? We converted extra carbohydrates that we get from the diet to the fatty acids and store as a triglyceride. And in which tissue, in which physiological state does lipogenesis occur? Fat fasting, fat, yes, in fat condition, right? When only we have, particularly when we have eat something, high carbohydrate diet, right? And we converted extra carbohydrate that we don't need, or we can, burn whatever, oxidize into the fat. Question, yes. Yes, that's good question. I think compared to the protein, carbohydrate is more strong drivers, for lipogenesis, right? Because protein has a little bit different function, right? And mostly we need a protein to build the tissues, and the other, or to make hormones and enzymes, right? This need to be refreshed, so the different function of protein. So again, carbohydrates more strong drivers or stimuli of lipogenesis. And where does lipogenesis take place in the cell? What do you expect? Cytosol, mitochondria, or somewhere else? Cytosol, right? It's considered the synthetic pathway, right? So again, lipogenesis occurs in the cytosol. And last point, what are the building block molecules of fatty acids? You mentioned many times, right? The building block molecules, or the starting molecule, substrate. Yes, acetyl-CoA. So acetyl-CoA is the building block molecules, right, of fatty acids. So again, fatty acids is synthesized from acetyl-CoA, right? So, all right. And just some, pretty much similar like we just described, right? So again, de novo fatty acid synthesis or lipogenesis is a process that converts surplus glucose or its metabolite intermediate to fat or fatty acids, more specifically, right? And precursors, although I put a glucose pyruvate lactate here, but I wanted you to remember that acetyl-CoA, right, is the most important molecules. Glucose pyruvate lactate eventually will all to be converted to acetyl-CoA, right, oxidized acetyl-CoA. And acetyl-CoA is, again, it's a building block molecule for fatty acid synthesis. And starting molecules, again, acetyl-CoA, right? So end product of a lipogenesis is the fatty acids, right? And I just put the palmitate, which is a 6-carbon saturated fatty acids, right? Of course, our body can also synthesize the other like 18-carbon fatty acids, or even unsaturated fatty acids, right? Some unsaturated fatty acids. But most active, the process is synthesized palmitate. So palmitate, our body can synthesize a lot. And again, the cellular localization of lipogenesis is in the cytosol, and primary tissues for lipogenesis are liver and adipose tissue, right? a nutritional state, where does the lipogenesis occur? Mostly actually in the well-fed, particularly carbohydrate diet, right? The unfed condition. Okay. So this is sort of a diagram that I put all the process that I described, right? In the diagram, I hopefully will show you, better the lipogenesis process. So first, high carbohydrate diet, right? Is driver of the lipogenesis. So when we eat a meal, particularly high in carbohydrate, we raise a very high levels of blood glucose, right? And this will trigger insulin secretion, right? We talked a lot in the last session, and also increase the some substrate levels like glucose, right? So we have, after the high carbohydrate meal, we have a high insulin levels and also glucose levels, right, in the circulation, and that will cause the I just wanted to remind you again, right? Glycolysis and glycogen synthesis pathway very active, right? To store glucose that we cannot be used, right? Glucose cannot be used in the liver and also in the muscle, glycolysis and glycogen synthesis. And in addition to these very active pathways, and our body can also convert extra glucose that cannot be used or stored to the fat. So glucose being converted to fat, and this is the lipogenesis process, right? So this primarily occurs in the liver. So lipogenesis, and then fatty acid synthesized from glucose, right, in the liver, and be packed into the one more step. So fatty acids, once synthesized, and then be converted to triglyceride, right? And triglyceride synthesized initially from glucose. Liver is not the main storage site for triglyceride, right? So adipose tissue is the storage site of triglyceride. So triglyceride need to be transported from the liver to the adipose tissue and stored in the adipose tissue in the fat condition, right? So this process again involves the lipoproteins, VLDL particularly, right, is synthesized in the liver. We don't worry about the lipoprotein now, but we will talk about it later. So triglyceride being packed into the VLDL lipoprotein and be transported to the adipose tissue. And muscle can also do some, lipogenesis, and adipose tissue is also takes up fatty acids, right? Take up the glucose and convert it to fatty acids. So lipogenesis can also take place in the adipose tissue, but relatively, small compared to the liver. And the other thing that I wanted to mention to you is that the triglyceride that transported from liver to adipose tissue involve the very important process. It's called that involves the lipoprotein lipase, LPL. The reason is the triglyceride in the VLDL, right, is relatively large molecules compared to fatty acids, right? So this triglyceride, because of too large, that cannot be uptake by adipose tissue or adipocyte. So need to be break down to fatty acids And then free fatty acids then can be uptaken by fat tissue. So lipoprotein lipase is the enzyme that can break down triglyceride and release fatty acids and glycerol, right? All right, any question about this lipogenesis process, right? So again, fatty acids can be synthesized from glucose, the extra glucose in the liver, but these fatty acids need to be stored in adipose tissue, right? So be synthesized triglyceride packed into lipoproteins and transported into fat tissue for storage. All right, so now let's get to the details, right? We have a big picture, right? And how lipogenesis take place. Now get the details. So synthesis of fatty acids. This is just the overall pictures, right? How palmitated fatty acids, 6-carbon saturated fatty acids, synthesized from acetyl-CoA. And this requires two main enzymes and also other substrates like NADPH, if you still remember when we talk about pentophosphate pathway, right? And one of the function of pentophosphate pathway is to synthesize NADPH, and this is required for fatty acid synthesis, right? So NADPH is required, and also some metals, and biotin, right? And bicarbonate are all required for the synthesis of fatty acids, and the two enzymes complex involved, right, or regulates the synthesis of fatty acids. One is acetyl-CoA carboxylase, and the second enzyme complex is fatty acid synthesis complex, right? So acetyl-CoA carboxylase is a key enzyme, right, that control lipogenesis rate. So try to remember acetyl-CoA carboxylase, or we call it ACC, right, as the key enzyme that carry the key steps of the lipogenesis. So now I'm going to introduce the citrate shuttles. If you still remember the TC cycle right here, this is a mitochondria, right? And this is a cytosol, let's say. We just talked about the location of lipogenesis in the cell, in the cytosol, right? So lipogenesis occurs in the cytosol, where is acetyl-CoA generated in cytocells, which cellular compartment the acetyl-CoA is generated. In the mitochondria, right, is converted from pyruvate. So, but lipogenesis occurs in the cytosol. So acetyl-CoA needs to be moved from mitochondria to cytosol, right, for lipogenesis. And this is, again, the mitochondria and the TC cycle, right? And I just put a more, connection to the glucose and amino acids. So both glucose and amino acids can be metabolized to pyruvate, and pyruvate then enter the mitochondria, right? And for this reaction, for further oxidation, right? Pyruvate to acetyl-CoA. So acetyl-CoA, again, is generated from pyruvate by pyruvate dehydrogenase in the mitochondria. And so fatty acid synthesis starts from acetyl-CoA, right? Acetyl-CoA then once it's generated in the mitochondria, then enter the TCA cycles, if you still remember, right? The first reaction in the TCA cycle is the formation of citrate from acetyl-CoA and oxaloacetate, right? And citrate needs to be transported out of mitochondria to cytosol, and there is the shuttle or transporter, right, that can transport the citrate out of mitochondria to the cytosol, and this the T represented the tricarboxylate transporter, right? So tricarboxylate transporter basically transports citrate out of the mitochondria to the cytosol. And once it gets to the cytosol, the citrate can be break down to acetyl-CoA, back to acetyl-CoA and oxaloacetate. And oxaloacetate can be recycled, right, and back to the mitochondria, to TCA cycle. And acetyl-CoA, and this is the enzyme that breaks down citrate in the cytosol. It's ATP citrulase, right? ATP citrulase is the enzyme that breaks down citrate and back to acetyl-CoA. So acetyl-CoA eventually enter the lipogenesis pathway, right? So again, the purpose of citrate shuttles is to facilitate acetyl-CoA to move across the inner mitochondrial membrane to the cytosol, right? So in general, I mean, this citrate purpose is to move acetyl-CoA to the cytosol, but the details that involve the citrate, right? So the real molecule that move out of the mitochondria is a citrate. So acetyl-CoA need to be converted citrate first, and then be transported into cytosol and be break down, back to the citrate. The acetyl-CoA, right? So try to remember what is the purpose of a citrate shuttle, right? Now this is the first or initial controlling step of lipogenesis. That is the formation of marino-CoA. So acetyl-CoA once is arrived in the cytosol, then the first step is the acetyl-CoA being converted to marino-CoA, right? So marino-CoA formation again is the key first reaction of the lipogenesis pathway. And this is again the key enzyme, the acetyl-CoA carboxylase, ACC, is the key enzyme that control the formation of marino-CoA from acetyl-CoA, right? And ACC actually is the complex of enzymes. That means the several enzymes, right, involve the ACC activities. So it's not just the one enzyme. So basically, it's a multi-enzyme protein that has three enzyme activities. One is biotin carboxylase. A second one is biotin carboxyl carrier protein, right? And last is trans-carboxylase. So this protein that come very close and work together, right, to catalyze the formation of a marino-CoA. So in general, the two steps of reaction that involve the formation of marino-CoA. First is the step, right? The first step is the step that catalyzed by biotin carboxylase. The enzyme biotin basically is the biotin carboxyl carrier protein, right? So first, the carboxyl group that initially from bicarbonate being transferred to biotin, right? So this is first reaction and catalyzed by these two protein or enzymes. And the second step is the carboxyl group that from biotin, enzyme biotin, being transferred to acetyl-CoA molecules, right? So you're adding one carbon to the acetyl-CoA molecules. So acetyl-CoA molecule basically has 2 carbons, right? And malonyl-CoA has 3 carbons. So, and this 1 carbon donors is donated from bicarbonate, right? That makes sense. So eventually you form the malonyl-CoA that has 3 carbons, right? If you compare to 2 carbons of acetyl-CoA. Again, the carboxyl group, 1 carbon is from the initially bicarbonate. So try to remember, the substrate and enzymes that involve this first very important reaction of malonyl-CoA, right, formation, the apogenesis. And secondly, once the malonyl-CoA is formed, that's the synthesis of fatty acids continues. So the next enzyme complex is fatty acid synthesis. This complex is even more complicated, right? Basically has the 2 called a monomer, right? Actually, these are 2 identical enzyme complex. So each monomer contains 7 enzymes, right? So they just like copy, but they arrange in the opposite direction. So, but you don't have to remember each enzyme, but just it's very complex, right? The enzyme contains double, actually 7 enzymes, but double, right? So, so each this kind of arrangement and makes the enzyme has 2 end. One is called a cis SH end, and the other one is pan SH or soft hydroxyl group, right? So this basically can this 2 sides or end basically you can be attached by the acyl group. So it's a place that you can extend the hydrogen carbon chain of fatty acids. And before we talk about the details, I wanted to clarify the concept. Acetyl-CoA versus acyl-CoA. These are actually the 2 different molecules, right? Acetyl-CoA I just mentioned only has 2 carbons, right? And a CoA group. This is malonyl-CoA. Malonyl-CoA has 3 carbons, right? And acyl group. And acetyl-CoA has 2 carbons and acyl group. And acyl-CoA, acyl group and R group. R group basically is the carbon-hydrogen chain, and it depends, depends on what type of fatty acids. If the palmitate CoA, that means the has 16 carbons, right? And if the other type of means that the R group of carbon chain can be varied, depending on the fatty acid, whether it's 16 carbon fatty acids, 18, 20, or whatever, right? So acyl-CoA basically is the long chain, very long chain fatty acids with a CoA group attached, right? So and also when you see this name, different name, actually the same thing. Sometimes we call it fatty acyl-CoA or long chain acyl-CoA or acyl-CoA, right? Basically the same talk about the same thing, right? Okay, so it's very complicated process of a fatty acid synthesis by fatty acid synthesis complex, right? But we will try to make things simple, right? So you try to remember. I don't know, should we take a break? All right. Okay, take a break and then we come back to talk about it. Okay, are we ready? All right. So, it's a really busy slide, right? So the fatty acid synthesis complex that involved process is, first of all, it's kind of cycling, right? The fatty acids, we know fatty acids are long carbon chain, right? And here we're just taking the palmitate as an example, how palmitate, which has a 16 carbon fatty acid, is being synthesized from 2 carbon acetyl-CoA, right? So, so each cycles that you adding 2 carbons, right? And then you go in cycles, and each cycle you extend the carbon chain 2 carbon longer, right, to making the acyl CoA with 2 carbon longer, and involves several reactions in each cycle, right? So again, the first cycles that requires the acetyl-CoA, 1 acetyl-CoA molecules and 1 marino-CoA molecules, right? And to form 4 carbon acetyl-CoA chain, 4 carbon acyl chain, right? And the subsequent cycles, starting with an acyl chain 2 carbons longer, right? And 2 carbons added to existing acyl chain, right? Each cycle, and 2 carbon unit basically is donated from, remember, marino-CoA, not as you expect acetyl-CoA, right? And lastly, the 2 NADPH molecules are needed as the carbon donor, as the hydrogen donors, right? Used in each cycle. Okay, now let's start from the beginning. So, you guys able to see? Probably in your notes, right? Not here. Yes. So again, this represents a complex of a fatty acid synthesis. Remember, I told you, the 2 monomer, right, form the complex and has the 2 different ends. One the cis SH group, and the other one has the pan SH group. And this 2 side actually is very important. That is acetyl-CoA molecules or marino-CoA molecules attached to this cis or pan SH side, right? So first, this is the first reaction, right? The key reaction, we already talked about it, right? The acetyl-CoA is converted to marino-CoA by ACC, key enzyme. This is a key reaction. And once the malonyl-CoA is synthesized, so malonyl-CoA molecule be attached to the pan-SH end of the fatty acid synthesis complex, right? And one molecule of acetyl-CoA being attached to cis-SH end, right? So this is the acetyl-CoA, and this is the malonyl-CoA. And then once the let's see, and then the next step is the loss of the one carbon of a carboxyl group from let's see, from actually the malonyl-CoA, right? So this carboxyl group is lost, and then acetyl-CoA group is transferred from cis-SHN to this malonyl-CoA molecules, right, at the pan-SH. So carboxyl group is lost as the CO2, right, released. So eventually you see the forming a carbonyl group, right, at the end of the malonyl-CoA molecules. Right. So this group called a ketoacyl enzyme, right? Basically the ketoacyl group that attached to the fatty acid synthesis enzyme at the pan end. Right. Now the next step is the reduction reaction that requires NADPH, right? One NADPH. So NADPH, the proton actually is added to the carboxyl group and converted it to the hydroxyl group, right? So this proton basically is from the NADH, and this is a reduction reaction. And next, the loss of water. The carboxyl group, the water is lost, and then forming the double bond, right? So unsaturated acyl enzyme, so unsaturated acyl group that attached to the enzyme, and again also at the pan side, right? And lastly, the additional reduction reaction, also this group reaction requires NADH, and then adding the proton to remove the double bond, right? And then form the 4-carbon acyl-CoA group, right, attached to the enzyme. So this is the first cycle of the fatty acid synthesis, and again requires the one acetyl-CoA molecules and one malonyl-CoA molecules, and 2 NADPH for the reduction reaction, right? And then the second cycles or the subsequent cycles, that actually this acyl group then be added to the cis-SH end instead of acetyl-CoA group, right? So and malonyl-CoA will continue to require malonyl-CoA for the subsequent cycles. So malonyl-CoA was still adding to the pan ends, right? And doing the same reaction again, so going the cycles. So again, each cycles you're adding the 2 carbon, so extend acyl-CoA chain with the 2 carbons longer, right? Does that make sense? Again, the carbon donors is the malonyl-CoA, not the acetyl-CoA, all right? So now I have a question. So how many cycles that we need in order to form or generate the 6 carbon palmitate fatty acids? So first cycles, we form 4 carbons, right? Acyl-CoA chain, and then we go, each cycle we're adding 2, right, carbons, so extend the 2 carbons. So how many cycles is needed? No. Pentotate 16 carbons, right? 7, yes, 7, right? So 7 cycles is needed, right, to complete the synthesis of 16 carbons pentotate, right? So again, what you need to remember is try to thinking this is the cycling, right? The synthesis of fatty acids is going cycling, and each cycle is adding 2 carbons, and substrate for the fatty acid synthesis cycles is acetyl-CoA, right, and malonyl-CoA. And in terms of numbers, how many acetyl-CoA, how many malonyl-CoA needed, you guys can thinking about it, and we have something to talk about later. So this just show you the pentotate fatty acids molecules, and the carboxyl end and methyl end. Basically, the methyl end are 2 carbons, right? From the methyl end is initially from acetyl-CoA, and the rest of the carbon actually from malonyl-CoA. Right? So just give you some idea. Again, the carbon donor, 2 carbon donors is malonyl-CoA. And provision of acetyl-CoA and NADPH. So again, acetyl-CoA is the starting molecule, right? So where does acetyl-CoA comes from? We talk about, right? Carbohydrate metabolism, right? Glucose and oxidation glucose and amino acids, right? So acetyl-CoA that being utilized for fatty acid synthesis basically is from oxidation of glucose and amino acids. And what about NADPH? One pathway, right, is generally NADPH we talk about, right? Pentose, phosphate pathway. And the other 2 sources that we have not talked about it, Maillard enzyme reactions and isocitrate dehydrogenase. So, you can just see that here is the one sources of NADH and Maillard, right? And citrate dehydrogenase is another source of the NADH. So again, NADH is coming from 3 sources. One is the pentose phosphate pathway, and second source is from the Maillard enzyme catalyzed reaction, right? And third is the isocitrate dehydrogenase catalyzed reaction. Okay, now something you guys need to do is counting for substrates, right? For synthesize a pentotate, how many acetyl-CoA molecules are needed? 1, yes, 1 acetyl-CoA. And how many malonyl-CoA? 7, yeah, 7, because going 7 cycles, right? Each cycle you need 1 malonyl-CoA. And how many NADPH? How many NADPH is required for each cycle?
2.
Yeah, so then total 14, right? 14. Okay. Now we just finished the synthesis of a saturated 16 palmitate synthesis, right? And starting from acetyl-CoA molecules, and first key reactions is the formation of a malonyl-CoA from acetyl-CoA. What is the key enzyme for first key reactions? You guys remember? What is the key enzyme? Just go back to your notes. Yeah, acetyl-CoA carboxylate, ACC, right, is a complex, and this complex has a multiple enzyme, right? Try to remember, and then involve the 2 steps, right, for malonyl-CoA formation. It's kind of complicated, right? You're only just adding 1 more carbon, and you require the multiple steps and enzyme involved. So, and then the next step is the fatty acid synthesis enzyme cycles, right? And try to remember, how each cycle requires the donors, carbon donors. What are the 2 carbon donors? What is the carbon donor? 2 carbon donors for fatty acid synthesis? Malonyl-CoA, right? And proton donors, NADH, right? And sources of NADH, 3 sources. Pentophosphate pathway, right? Maillard enzyme and isocitrate dehydrogenase. Okay, now let's talk about the additional kind of process that require for synthesize fatty acids that longer than 16 carbons. So, for example, like 18 or 20, right, the carbons, and what our body do is usually just do the elongation of a fatty acyl-CoA, right? And the process is pretty similar, like we just described in the fatty acid synthesis process, right? Also go in the cycles, depending on how many carbons that needed, to be extended, and the process occurs in the ER, right? It's in the cytosol, but close to ER, and start with the long chain acyl-CoA instead of acetyl-CoA, right? So elongation process, you start with the long chain acyl-CoA as a substrate, and the reaction goes similar. You have 2 reduction reactions and condensation, and the same actually reaction that described in the fatty acid synthesis cycles, and the malonyl-CoA is also the carbon donors, right, for elongation reactions, and NADPH is also the electron donors, right? So similar, like but just the substrate is different, right? It's the long chain acyl-CoA, not the acetyl-CoA or malonyl-CoA. So that is kind of simple, I think. Yes.
Is NADPH an electron donor, or you just mentioned a little bit it was like a hydrogen?
Yeah, it's pretty similar, I think. The function, because the proton also carries like electron, right? So sometimes we call the electron not proton. Pretty much similar, I think. Okay. Now, how about some fatty acid that has the double bonds, right? Like unsaturated fatty acids. How we synthesize unsaturated fatty acids. So for the monounsaturated fatty acids, that means the fatty acid only has a 1 double bond, right? And this process requires an enzyme called a desaturase. So our body has the certain type of desaturase, so can synthesize monounsaturated fatty acids. And here, just give you one example, like saturate the palmitate, right? The can be converted to the palmitoyl-CoA, which has a 1 double bond at the delta 9 position. So it's catalyzed by an enzyme called delta desaturase. So that delta 9 desaturase means that the enzyme catalyzed formation of double bond at the delta 9 carbon position, right? And the desaturation usually occurs in ER and particularly in the liver, right? And requires again the delta 9 desaturase, and substrate is the the palmitate. Okay, now how about the polyunsaturated fatty acids? Fatty acid has more than 1 double bond, right? And you have a longer carbon chain. Then 2 steps actually required. One is called elongation, and the other step is desaturation. So we need to extend the carbon chain, and meantime we need to catalyze the formation of double bonds, right? So we have delta 4, 5, and 6 desaturase, elongase, and to extend carbon chain and to form the double bonds, right? So I'm not going to go into detail, but just simple, right? The 2 steps you can synthesize polyunsaturated fatty acids. And here I just wanted to remind you on the numbering system, how fatty acids be numbered carbon, particularly the position, right? I hope you're not kind of confused. We actually use 3 different numbering systems. For example, the 1, 2, 3, this type of system, basically the name the carbons from the carboxyl carbon, right? The first carbon, named 1. So this is carbon number 1, and then 2, 3, and 4, right? Started from the carboxyl end. And the alpha, beta, gamma type of a system, basically you name the carbons adjacent to the carboxyl carbon. Usually this is the number 2 carbon named as the alpha carbon, right? And the beta and gamma. And omega system is actually goes opposite direction. This named the terminal methyl carbon as omega 1, right? And then 2 and 3. So sometimes you heard like a beta oxidation, fatty acids beta oxidation. We're going to talk about later. So basically, because you break the fatty acids at the beta carbon position, so we give a kind of a name called a beta oxidation. And also you heard like omega 3, omega 6 fatty acid, right? So basically that. So this is basically based on the double bond position, whether it's at the omega 3, start at omega 3, or start at omega 6, so that we name the the fatty acids. Now regulation of beta lipogenesis. This overall regulation, and we already talked about the 2 different type of diet, right? The carbohydrate diet definitely stimulate lipogenesis, and insulin that related to the high carbohydrate diet also stimulate lipogenesis, right? And high sucrose or high diet also stimulate lipogenesis. The high-fat diet, calorie restriction, or the glucagon basically inhibits lipogenesis. And the details of the regulation, again, I want you guys to focusing on the key enzymes, acetyl-CoA carboxylase, ACC, how ACC can be regulated, then can control the whole rate of the lipogenesis, right? So actually ACC is regulated at 3 levels. First is called allosteric regulation, basically regulated by the substrate, right? And second regulation, the covalent regulation, basically regulated by hormones that regulate enzyme phosphorylation or dephosphorylation, right? And lastly, the gene transcription, the gene expression. So let's talk first the allosteric regulation. The 2 key substrate are citrate and long-chain acyl-CoA, right? And these 2 substrate regulate ACC activity in the opposite way, right? So, for example, citrate basically stimulate ACC, right, and stimulate lipogenesis. And long-chain CoA serve as inhibitors of ACC, so inhibits the lipogenesis. So here is the reason that we talked about earlier, why high-fat diet will inhibit lipogenesis, because when we eat a high-fat diet, right, we have lots of fatty acids levels in the circulation. So fatty acids basically can be activated to the long-chain fatty acyl-CoA. So we have high levels of long-chain fatty acyl-CoA in the body when we eat a high-fat diet, and this will inhibit the ACC key enzyme for lipogenesis, right? So this is how high-fat diet inhibits lipogenesis, basically by raising the high long-chain fatty acid acyl-CoA levels, right, in the blood. So then can inhibit the ACC activity. That makes sense, right? And covalent regulation, basically insulin or glucagon can regulate phosphorylation, dephosphorylation of ACC, right? We have a more detail in the next slide. And lastly, the gene expression, right? Basically, insulin and glucose both can increase ACC gene expression, so increase ACC level, right? That will stimulate lipogenesis. And glucagon or cyclo-AMP, and these 2 kind of things pair kind of together, right? The fasting condition, we have a high glucagon and high cyclo-AMP. So both decreases lipogenesis, decrease ACC gene expression, right? Now I wanted to introduce you one important, very important enzyme that also involves the regulation of ACC. That is the called AMP, activated protein kinase, AMPK, right? AMPK is the enzyme, is a kinase, basically regulates phosphorylation and dephosphorylation of ACC here. Try to focus on this box, right? So AMPK, when it's activated, then can causing the phosphorylation of ACC. And this is also kind of the exception. Usually I mentioned, right, when enzyme or protein gets phosphorylated, the activity is increased, and only under the very rare kind of condition that the phosphorylated protein actually causing the decreased activity. And here is the like ACC. So when ACC is phosphorylated by AMPK, then ACC activity is decreased, so that decrease the lipogenesis, right? So when AMPK activity is increased usually during the starvation, right? When we have a low energy, and the epinephrine, glucagon, or cyclo-AMP levels goes up, then AMPK can be activated, right? And then phosphorylate ACC causing the decreased ACC activities and decreased lipogenesis, right? So again, we just try to add one more, enzyme, AMPK, how it regulates ACC and lipogenesis. Any questions? Right. And this is something that I wanted to mention again, that how long chain acyl-CoA inhibits lipogenesis, right? Again, high-fat diet causing increased long chain fatty acid levels, and we already mentioned a couple slides ago, right? The long chain fatty acid can directly inhibit ACC, but in addition to this mechanism, they are also involved the other two mechanisms, right? So first, again, long chain acyl-CoA can directly inhibit the ACC activity, right? And the second mechanism is that it inhibits the transporter activities. Remember the citrate shuttle, right? So long chain acyl-CoA can also inhibit the transporter of citrate into the cytosol. This is the second mechanism, how long chain acyl-CoA inhibits lipogenesis. And lastly, long chain acyl-CoA can also inhibit pyruvate dehydrogenase. This is the enzyme that catalyzes the formation of acetyl-CoA from pyruvate, right? And so three mechanisms, and the next slide I give you the more details, right? Put on together. So the long chain acyl-CoA can directly inhibit ACC activity, and also inhibits the transporter is citrate, right? And inhibits the pyruvate dehydrogenase and causing the decrease, the substrate, right? Any question? Okay. I know lots of information, right? So, okay. So, if we're just thinking about insulin regulation lipogenesis, in general, what do you think insulin should regulate lipogenesis? Up or down, or increase, decrease lipogenesis? Increase, right? Basically. So, and so insulin. First, insulin can stimulate ACC activity, right, and control the first reaction of lipogenesis. And secondly, because of insulin can also increase glucose uptake, right, and that will increase the production of pyruvate, and also increase the glycerol-3 phosphate. And insulin also increase the hydrogen pyruvate dehydrogenase. Sorry. And decrease the intracellular cyclo-AMP levels because of insulin inhibits lipolysis, right? Okay, so the net results of insulin action on lipogenesis is increased lipogenesis, right? Okay, now I think I just want to summarize fatty acid synthesis or de novo lipogenesis. When does this occur in the fat condition, right? Particularly when a high carbohydrate diet. And the purpose of this process is to convert surplus glucose and also amino acids, right, to fat. And fatty acid synthesis basically occurs in the cytosol and requires acetyl-CoA as the building block molecules, right, or starting molecules. And two enzyme complexes that involved. First is the ACC complex, and this complex catalyzes the key reaction, right? Basically, is the formation of malonyl-CoA from acetyl-CoA, and the second complex is the fatty acid synthesis, and again is the going cycling, and each cycles added 2 carbons, and carbon donors is the malonyl-CoA, and NADPH is a proton donor. And in terms of regulation, try to remember the 3 levels, right? The allosteric regulation, citrate basically stimulates, and long-chain acyl-CoA inhibits ACC activities. And also we mentioned the AMPK, right? The kinase is upregulated under the fasting condition and inhibits the ACC and inhibits lipogenesis. All right, now let's talk about any questions about lipogenesis. Okay, now let's talk about triglyceride synthesis. Right, requires 2 substrates, the glycerol backbone, and 3 free fatty acids. Why triglycerides need to be synthesized or stored? Why we need to synthesize triglyceride? Yeah, for energy, right? Of course, but why we just store the fatty acids and not the triglyceride? Okay, so because of fatty acids is kind of active, right? It can somehow regulate the lot of metabolic activities. And triglycerides is more kind of natural, right? So we don't want to causing kind of unnecessary regulation, right, or stimulation, whatever. So we need to store the converted fatty acids to triglyceride, the natural lipids, and then store in the cell, fat cell, right? And where are triglycerides synthesized? Which tissues does the most activities of triglyceride synthesis? Yes, adipose tissue. So it's different from the lipogenesis, right? Lipogenesis basically liver does the most important role, and the triglyceride synthesis is basically mostly active in the adipose tissue. But of course, liver can also synthesize triglyceride, right, and muscle as well. So, and in the cells, which part of cells that does the triglyceride synthesis? It's also in the cytosol, but facing the ER and facing the cytosol, right? So again, the triglyceride basically synthesize ER. And that are facing the cytosol. And storage, of course, you guys, everybody knows, right? Stored in the adipose tissue. But some of triglycerides can also be stored in the liver and also the muscle, right? But we don't want to have lots of triglycerides stored in the liver, right? And causing liver disease. So, in a normal condition, physiological condition, we have kind of a small amount of triglyceride. So the process of the triglyceride synthesis. So fatty acids, first of all, need to be activated before they can be attached to the glycerol backbone, right? So this reaction is called fatty acid activation. Basically, you attach the acyl group to the fatty acids, forming fatty acyl-CoA, right? So fatty acyl-CoA is the molecules, fatty acid molecule that can be attached to the triglyceride backbone. And this is enzyme, fatty acyl-CoA synthetase as an enzyme to catalyze the activation of fatty acids to fatty acyl-CoA, right? So this is the first reaction. And we also need a glycerol backbone, right? And glycerol 3-phosphate as a substrate. I think we talked about this early, right, in the carbohydrate unit. If you still remember, one of the source, no? And then you have to come back next year. So remember, we talk about the adipose tissue and how carbohydrate being metabolized in the adipose tissue. One of the important process is that glucose can be metabolized and generate glycerol 3 backbone, right? So glycolysis and gluconeogenesis, two pathways that can, particularly glycolysis, can generate glycerol 3 backbone from glucose. Remember this, right? So this is one of the important source of a glycerol 3 backbone. And the other source is the glycerol 3-phosphate can be directly generated from glycerol. That requires an enzyme called glycerol kinase, right? And glycerol kinase, remember, only present in the liver. So this sources of a glycerol 3-phosphate only can be generated in the liver and not in adipose tissue, right? So adipose tissue, again, basically, in adipose tissue, glycerol 3-phosphate is generated from glycolysis from glucose. And in the liver, that the glycerol 3-phosphate can be generated from two pathways, right? One is the glycolysis, and the other pathway directly from the glycerol by glycerol kinase, right? Now, what about the fatty acids? Where does the fatty acid come from when we need to synthesize, triglyceride? And usually triglyceride is synthesized in which physiological condition? What do you think? Fat, right? Fat condition, we need to store energy. So in fat condition, when we eat, we usually get fatty acids from the diet, right? So fatty acids from the diet is one of the source of fatty acids for triglyceride synthesis. And a second, de novo lipogenesis, remember, right? We eat not fat, but also carbohydrate, protein, and extra carbohydrate protein can be converted to fat by lipogenesis in the liver, right? So this can also contribute to fatty acids for triglyceride synthesis, right? So second sources of fatty acids from lipogenesis. Does that make sense? And lastly, body stored means that we also be able to release fatty acids from stored triglyceride. And this usually occurred under sort of a pathological condition, like, for example, like a diabetes, right? Like diabetic obesity conditions, usually we have a high rate of lipolysis. That means we have a release a lot of fatty acids from the fat tissue, right? And release the fatty acids into the circulation. So basically, obese and particularly type 2 diabetic patients have a high levels of fatty acids in the blood. And this high level fatty acid can also serve as the substrate of triglyceride synthesis. So liver can pick up this fatty acids, which initially comes from fat tissue, right? And pick up into the liver and synthesize triglyceride and store in the liver. So that is sort of the mechanism for, one of the mechanisms for the fatty liver disease, you know. So usually like obese patient or type 2 diabetic patient, very common that have fatty liver disease. Again, because of they somehow have high levels of lipolysis, so high levels of fatty acids in circulation, and liver picks up, right? You know, somehow the fat tissue loses its function to store lipids, so they release lipids, and the liver picks up, or even some other tissue like muscle can also pick up fatty acids and synthesize triglycerides and store in the non-edible tissue, right? Okay, so just try to remember the fatty acids for triglyceride synthesis. Fatty acids are coming from 3 different sources. One is diet, and second one is the de novo lipogenesis, right? And the third is again from the stored lipids or triglyceride, basically under the disease or pathological condition, right? Any questions? Okay, for the synthesis of triglyceride, there are several steps, and you just understand it, right? You don't have to remember each step, what enzymes are involved or something like this. So very simple. I mean, you just need a backbone, right? And then you're just adding the 3 fatty acids to the backbone. So adding each fatty acid to the specific, you know, the carbon position of a glycerol backbone that requires the enzyme, right? So the first, we need generate the 3 phosphate, and I mentioned like 2 different sources of glycerol 3 phosphate, right? One is from glycolysis, and the other one is from directly phosphorylation of glycerol, right? And the first reaction is adding the first fatty acids to the SN1 position, right? And by this enzyme glycerol 3 phosphate acetyltransferase. And usually the first fatty acids that adding to the SN1 position is the saturated fatty acids, mainly the saturated fatty acid, right? And the second step, you're just adding the another fatty acids to the SN2 position, right? And this fatty acids adding to SN2 position, basically unsaturated fatty acids, right? Mostly unsaturated fatty acids. So you generating the diacylglycerophosphate, right? You have already have two fatty acids being added to the SN1, SN2 position of glycerol backbone, and you still have a phosphate left, right, at the SN3 position. So the next step, before you adding the third fatty acids to the SN3 position, you need to remove the phosphate first. So then removal of the phosphate from the diacylglycerophosphate, and you generate diacylglycerol, right? So diacylglycerol basically is a glycerol backbone, has a glycerol backbone, and the two fatty acids attached to SN1 and the SN2 position. Okay, so this is the last step, right? You adding the last fatty acids to the SN3 position and generating the triglycerol. And the enzyme, the diacylglycerol isotransferase, DGAT, this is a key enzyme, right? Control the synthesis of triglyceride, right? So DGAT is the key enzyme in the triglyceride synthesis pathway and catalyze the last fatty acids adding to the SN3 position, right? Okay, so I think it's pretty simple, right? For triglyceride synthesis, just try to remember the substrate, the sources of the substrate, particularly glycerol 3-phosphate, right, coming from two pathways, glycolysis and the glycerol phosphorylation, and you just adding the three fatty acids. And again, before fatty acids being able to add it to the glycerol, fatty acid need to be activated, right, to fatty acyl-CoA by fatty acyl synthetase. And last step, the DGAP, is a key enzyme, right, to catalyze the. And transport. So once you synthesize triglyceride, I'm talking about here, the synthesized triglyceride is not in edible tissue, in the liver, then triglyceride need to be transported to the final destination, right? Because triglyceride usually stored in the edible tissue. So again, the synthesized triglyceride in the liver need to be transported out of liver and to the edible tissue for storage. But again, the triglyceride cannot be just simply transported to the fat tissue, but requires a complex, very complex protein called lipoprotein, right? We have specific session talk about lipoproteins. So lipoprotein, that is a protein complex that carries the triglyceride from the liver to the adipose tissue, right? And specific lipoprotein that can be synthesized by the liver is called a very low density lipoprotein, the VLDL. But triglyceride, once is absorbed from the diet in the small intestine, we have, I think we talked about it, right? If you still remember, it's carried by a different lipoprotein, which is chylomicron, right? So chylomicron is synthesized in the small intestine and basically carry the triglyceride from small intestine to the adipose tissue for storage, right? And just go back to this slide. You know, again, when we eat and digest the lipids, and we absorb the fatty acids and resynthesize fatty acids again in the enterocyte, the small intestine, if you still remember, right? And the enterocyte can also synthesize chylomicron, so that it can carry triglyceride from small intestine to the adipose tissue, and some of the chylomicron can also, transport the triglyceride to the liver. But majority of triglyceride being transported by chylomicron to the liver. To the fat tissue. And in the liver, because of lipogenesis, is very active, right, in the fat condition. And also have the triglyceride synthesized from glucose, and that had be packed to the LDL and LDL that carries the triglyceride from the liver to adipose tissue. And both triglyceride in both chylomicron and VLDL required to be break down by LPS, lipoprotein lipase, right, to release free fatty acids, and only free fatty acids can be uptaken by a adipose tissue, right? So once fatty acids goes into the fat tissue and then need to be resynthesized, triglyceride and stored in the adipose tissue. So the triglyceride synthesis activity is very active in the adipose tissue, adipocyte, right? Is it clear? Any questions? Okay. I think maybe we'll just stop here, and next time we're going to talk about, lipoprotein lipase, how they does function.